Hello, everyone. Thanks for tuning in to the Issue Podcast on the ASGCT Podcast Network. I'm your host, Emily Walsh-Martin, and today we have a very special guest on the pod, Federico Mingozzi. Are we incredibly lucky because Federico's first ASGCT podcast could have easily been on giants of gene therapy? Probably. Am I psyched that he could join us for an in-depth chat about his career journey and AAV immunogenicity? Absolutely. For those of you who don't know Federico, he has done seminal work throughout his career in AAV gene therapy, and his 2020 molecular therapy review of our current understanding of the interplay between these therapies and the immune system is a must-read for anyone trying to get a handle on the very complicated landscape of immune reactions to viral vectors. Will we solve all the challenges in the field over the course of the podcast? Definitely not. But will you leave enriched in your understanding? I sure hope so. Thanks for joining us. But before we get into the conversation, I want to remind everyone that the ASGCT annual meeting will be here before you know it. Meet us in Baltimore, May 7th through 11th for the only must-attend event in cell and gene therapy. Keynote speakers this year include Doctors Beverly Davidson, Charles Murray, Kevin Campbell, and Philip Gregory. There's no better place to deep dive in this field than the ASGCT annual meeting. You will not find the same breadth or depth of cell and gene therapy research anywhere in the world. Now entering its 27th year, the ASGCT annual meeting is the premier event for professionals in our field, and ASGCT members receive some truly incredible discounts on registration. So renew your membership and sign up for the meeting at asgct.org. Can't wait to see you there. Hi, Federico Mingozzi. Welcome to the podcast. Thanks so much for joining. You and I first met on a panel at this past year's ASGCT annual meeting. And one of the things that struck me was that we've both had very distinct career paths that led us to gene therapy drug development. Could you tell us a little bit about your journey?
Yes, absolutely. And hi, Emily. I'm very happy to be here today, and it's a great follow-up to that moment when we first met at the ASGCT. So my path in gene therapy, I would say I started my career in science in Italy. I studied at the University of Ferrara all the way to PhD, and there I was working on the genetics of coagulation factors. And nothing was really related to gene therapy, but in fact, the fact that I was working on coagulation factors was a link to my postdoc. In fact, in 2000, I moved to the United States as a postdoc, working with, at the time, Roland Herzog. And we started working on gene transfer with AAV vectors to deliver. And we were actually studying the induction of immunological tolerance to proteins expressed in the liver, and particularly clotting factors. That was my first time with gene therapy. And then after my postdoc, I continued to work on gene therapy. This time I was working with Kathy High, still at CHOP. And we started to, it was the first time really that I was exposed to the idea of bringing a gene therapy all the way to the clinic. And it was an exciting journey. I was there until 2013. And during that time, we were working not only on the development of gene therapies for hemophilia, and also we were working also with Jim Bennett and developing the gene therapy for Leber's congenital amaurosis, which then later on became an approved product, Luxturna. And so that was the time then truly we were facing the challenges of bringing a therapy, a new therapy, a gene therapy, all the way to patients, along with the study of the immune responses encountered in gene therapy which is a big chapter of my career and also still a very important point and hurdles of viral gene therapies. And then that was really a fantastic period and a highlight of my early career in science in academia. After that, I stayed in academia. I actually moved to France to become an independent scientist. We had a research group at Genethon, which is a research institute outside Paris. And I was also part of the INSERM, which is the equivalent of the NIH, the American NIH. And there I continue my work in gene therapy as well as immunology. And we brought to the clinic gene therapy for a rare liver indication called Crigler-Najjar syndrome. And that was my career in academia until I remember was the European Society of Gene Therapy meeting in 2016, I believe, yes. And I met again Kathy Hai, and she was talking about this company, Spark Therapeutics, and how there was the opportunity to join the company. And that's what happened. In 2017, I joined Spark as the chief scientific officer, and I was there for a while, for more than 6 years, first as a CSO, then a chief science and technology officer, until about a month ago when I started a new job.
Right. So you started a new voyage right now. So you're the CEO for a biotech, which is still in stealth. What are you looking forward to with this new adventure?
With this new adventure, I think there's multiple things I'm looking for. The first is I am a scientist by training, and I'm looking forward to learn what it means to be a CEO of a company. And sometimes I joke actually with people and I say, I have a lot of gaps in my understanding of what the job of a CEO is, to the point that I feel like they're canyons, they're not simply gaps. So there's a lot to learn and grow on my end. And as well as scientifically, because this new venture is still in genomic medicine, something that is very close to my heart, my scientific heart, and is a play with different technologies from what I've been doing for the past 20 plus years. So I'm looking forward to that as well. So there's this, I hope this will be a fantastic journey, both personally, professionally, but also scientifically.
Yeah. And maybe we can get the will he, won't he out of the way right now. I'm guessing you're not going to be able to yet share the name of your new venture. So we'll maybe just have to stay tuned.
Indeed. It will be a little while. At the moment, we decided that it's not the right time to disclose what's the name of the company, but eventually we will do so.
All right, maybe we'll have you back on the podcast to break the news. But before we get too distracted, let's go back to your career journey, because I think you've had a career journey that a lot of folks at ASGCT can relate to, because you've had that transition from academia to industry. And I'm curious, when you look back on that time, what were some of the biggest surprises for you in sort of making that move?
That's an excellent question. In fact, I must say one of the biggest, before I go into that, even one of the biggest drivers of my decision was to be in an environment that will allow me to do more for patients. So I experienced what it meant to bring a therapy from bench to bedside in academia. And that was a fantastic journey, but very limited by the resources available. And so I wanted to be in a context where I could do more for patients and do more at the same time, which is something also the idea of bringing a therapy all the way to the finish line is something that biotech is better suited than academia, although academia can still do a lot in terms of innovation, even clinical innovation. Then I joined Spark, and of course, coming from academia, I thought I knew everything I needed to know, and that was not the case. And in fact, I found myself surrounded by incredibly smart people who taught me a lot. And that was the first point that was very important for me. I learned quickly that drug development is very complex and requires a lot of skills that are not only the science skills. And I think that was something that in the academic environment I didn't completely grasp. And then the second point that I thought it was very eye-opening and very important is that when you're like an independent investigator, you run your team, essentially you are the sole decision maker most of the time. And that's not the kind of model of a company, or at least of a successful company, where in fact you need to join the brains of and the skills and make decisions that are usually reached through consensus and through analysis of the problem you're trying to solve for. And so those are some of the learnings that were crucial, I think, in my early days at Spark. And the last one, I would say, probably more relevant also, again, for a company, I learned how it's important to forget about the ego and rather be a good team player and think always for the best of the company and the patient and what you're doing.
Oh my gosh, so many things that you just said resonate so deeply with me. I think for me, my experience has also been that perhaps even more so than on the small molecule drug discovery and development side, gene therapy is a team sport. You're not going to get across the goal line without tapping a large diversity of expertises. And so, for me, what I always tell teams is reasonable people with the same information come to similar conclusions. And so I feel like if we can get all of the information out on the table and think it through, that's when we're best in a position to make good decisions that are going to help advance a program meaningfully for patients.
Yeah, I agree 100%. And the reason why this is particularly relevant for gene therapy is because gene therapies, I should use the plural, are complex and they're new. And oftentimes you're facing more unknowns than knowns. And so you have to make sure you have the right people around the table, and one person doesn't know everything necessarily.
Right. And the data sets are somewhat limited, right? Like the expanse of clinical exploration of small molecule pharmacokinetics and pharmacology is grand compared to the small numbers of patients who've had an AAV type therapy and the length of time we've been able to follow them. And so a lot of times we're trying to, I think, take the best information that's publicly available to help inform decisions, and it's still limited, that information. So we're having to really understand on a deep level why we're making certain choices as we develop these therapies.
Yeah, absolutely.
So, I feel like we're starting to get very close in our conversation here to the concept of immunology, because for me, that was always one of the biggest unknowns. And I will say, I use your 2020 molecular therapeutics overview as like required reading for all of my clients who are maybe it's their first time doing gene therapy, because I think you so eloquently lay out the complexities of the way that the immune system interacts with viral vectors. I'd love to know what your thoughts are about how much we've learned since you first started and also what's still out there to learn.
That's a great question. And by the way, that paper, it's interesting because I was very surprised to see is one of the most cited papers in the molecular therapy.
It's possible I'm single-handedly responsible for that, because I basically just hand it out to everyone.
I love that. Clearly, we've learned a lot. I mean, if I go back to the days these are the beginning of 2000, maybe 2004 or so. We were trying to understand what happened exactly in the first trial of liver gene therapy. It was a liver gene therapy for hemophilia B that we conducted at CHOP. And we had evidence that there was an immune response, but then things were really at the beginning, right? We were trying to understand which cell types were involved, and there was not a lot that we knew at the time. If you fast forward 20 years, today I think we learn a lot, and in particular, we learned that we're dealing with when it comes to AAV. One of the important players in what we see as far as immune responses is actually the early recognition of the virus by the innate immune system. In particular, we learned that we could design vectors that are a little less immunogenic than what we used to design by, for example, eliminating CG dimers from the genome. We learn about the role of complement, and that's still an open chapter. We also learned that the downstream immune responses involve CD8 T cells and all of that, right? And finally, we also learned that there's definitely a dose dependence in what we observe in the clinic with AAV, where definitely we can see some immune response that tend to be relatively benign in terms of clinical manifestations, so they tend to be asymptomatic and so on at lower vector doses. Of course, the risk there is that you will lose expression, so your therapy doesn't work, but there hasn't been severe or long-lasting detrimental outcomes in gene therapy trials. And then as we bump up the dose, as it was done for some trials, particularly in neuromuscular diseases, then we can encounter more acute toxicities, which seem to be complement-derived or related to the underlying condition, for example, hepatic toxicities and so on. So we learn a lot about the mechanisms behind these immune responses to AAV. And then we also learn more about how to address some of these immune-related issues. Part of it is using immunosuppression. I think the initial breakthrough, which was very, very simple, in fact, was when Amit Natwani and Andy Davidoff, so we're talking about UCL, University College London, and the St. Jude Children's Research Hospital. They started their AAV trial for hemophilia B. This time was an AAV8 vector, and basically what they did when they saw an immune response, an increase in liver enzymes, they gave steroids, and that worked, and was one of the first evidences that you could achieve long-term expression of a transgene in humans. Then over time, we learned that steroids work, but not all the time. So we also learn and are learning that perhaps in some cases, more complex immunosuppressive regimens are needed. Then I cannot talk about immune responses to AAV without saying that we are also always dealing with the fact that humans are exposed to AAV. And so we have immunity to these vectors. And one of the most limiting aspects of this pre-existing immunity is the presence of antibodies to AAV. And so that has been also, I work a lot on the topic in my past. And I would say having antibodies to AAV precludes the possibility to receive a gene therapy, whether that's the first gene therapy you will receive or the re-administration of the vector. And what is exciting is that while 20 years ago there was nothing to be done, now there's a lot of potential solutions also for that problem, whether that's the removal of these antibodies or, there's many solutions that have been developed in the preclinical space, with some of them soon to be applied in clinical trials. So I think in 20 years, we've done a lot of work, but also we've gone a long way This was a long answer, but
No, but what I love about the answer is I think you almost approach the answer in some ways, I guess how the field experienced it. So when, for instance, if anyone now like goes to read your 2020 molecular therapy paper, one of the things that you present very, very nicely is this concept that there's sort of three phases of the immune reaction, right? So there's the pre-existing immune component, there's the innate immunity that happens in the course of days, maybe a week, and then there's the sort of weeks, months adaptive response that occurs. And what I was struck with as you spoke is that, I think we as a field learned about these not in that order. We learned about these based on the fact of the tools that were available to us at the time. So we had assays for neutralizing antibodies or reasonable assays. And so that helped us to elaborate that part of the story. We had, later these, liver function tests. And then as you pointed out, the Nadwani experience, applying steroids after LFT elevation sort of taught us, okay, that's an immune part. I guess it strikes me that so much of our progress in understanding immunology has almost been sort of log step transitions as we get new sort of technologies that allow us to drive the inquiry better.
Yeah, I mean, absolutely, that's very true. And then the other point that is very important is that a lot of these immune responses are not predicted in animal models, right?
Right.
And so, and that was the true big challenge, and still is, right?
Yeah.
Basically, a lot of what we see happens only in the clinic. And as you were saying earlier, rightly, is that these are not trials of thousands of participants, right? These are small trials for rare diseases where we and others try to do their best to learn as much as possible about what was driving certain immune reactions and immune toxicities. It's not easy because, again, the numbers tend to be small, and it's very hard to go back and reproduce the findings in animal models, in most of the animal models.
And the reactions don't necessarily, it's not like every patient has the same
Correct.
type of reaction. And also, while we certainly, as you rightly pointed out, we see dose responsiveness to some of these reactions, that's also not, a perfect linear line either. So it does complicate, I'd say, truly understanding this, for sure.
It's very humbling, I would say, because and I'm a biologist by training, so I'm not a clinician. And I would say that on the research side, things are difficult, but you tend to work on models that are more or less controlled. And then the human system is the ultimate non-controlled system, right? Where you have genetics, environmental factors, disease-specific features, and which can be underlying inflammation, or there's a whole history that is hard to reproduce in a lab. And there's a whole uniqueness of the human immune system or the human body, which then results in a relatively high variability of outcomes.
Right. And there's a limitation to sort of how we can fully understand things. Like, so for instance, in hindsight, the fact that many of the initial sort of efforts in this space focused on hemophilia, they focused on factor IX, and that actually gave us an amazing handhold, because that's something that you can, very easily monitor over time and look at the impact and, the waxing and waning of the expression levels to try and understand, by adding the steroids, are we helping, are we hurting, what's going on? And so as you think about maybe some of the indication space that people are going after today, like it seems quite a bit harder to necessarily draw those correlates, right? For say a neuromuscular indication.
100%. That's a super important concept, which is the concept of what endpoints you're going to follow to understand or to guide your clinical decision, for instance, in managing immune responses. Hemophilia is the best. There's a few diseases that are similar, where you have a clear endpoint you follow, a clear biomarker that you follow, and you know that, if you are losing efficacy, and you know it pretty quickly, and so you can actually design an intervention. The issue for non-secreted proteins, like in neuromuscular diseases, or a protein that you're expressing in a closed compartment, let's say, is that you cannot follow that. And I would say even a lot of the immunological tests that we run, for example, an ELISpot, which is a common gold standard for immunomonitoring, it's hardly ever a good tool to make clinical decisions. And that's a big challenge. And so while you can still learn a lot, I think you're still facing the question of, what do I do? And generally, I'm going to generalize a little bit here, but I think it's something that has been done in the field, and I think it makes also sense, is that when you don't have a good biomarker that you can follow, then you try to design your clinical protocol in a way that you have you put in place some immunomodulation, let's say, that will prevent the immune response, so you don't have to worry too much about monitoring, because you don't have any way to monitor. And that could be also the case of, for example, if you're going to a disease affecting the CNS, where it's not like liver, there's very limited regenerative potential in the central nervous system, then you want to really try to design something where you maintain a good level of immunosuppression, you avoid inflammation, and you have to do it more thoroughly than, again, a hemophilia, where you can monitor expression, where a liver inflammation tends to be self-limited and not a big deal, because the liver will regenerate and so on.
Right. Yeah. No, I just remember early days, there were lots of debates, it seemed, about, how aggressive should one prophylax and the like. And it feels like, to your point, we're sort of landing in a spot where we're recognizing that, because this is one and done, meaning you have one shot on goal to provide the sort of best transduction and the best, chance at efficacy for these patients. There's something to be said for thoughtful, immunosuppression that reduces the likelihood that you're going to see things even from the beginning.
Yeah, and there's also another factor that I think is important. If we think beyond trials, and we want to put ourselves in a real life scenario where these therapies are actually approved therapies, then it's unlikely that we can have a system where you have a very, very close monitoring on every patient, because that's not the real life, right? So you want to be in a situation where you can prophylactically address these immune responses and not worry too much about monitoring the patient every minute. I think is more feasible, let's say, clinically.
Absolutely. Yeah. Let's shift gears just a little bit and talk about some of the other challenges that are, I think, still facing the field. And one of the ones that I think a lot about, I don't know if you'll agree, is the unknowns around how many cells do we need to transduce to have the best benefit, particularly for a non-secreted payload, right? And it feels like something where, in some cases, we're very lucky. We have sort of human genetics to maybe point us in a certain direction for, if you can correct, 20% of cells, you'll see a benefit and that sort of thing. But for some of these indications where it's not a secreted protein, like hemophilia, like how do you approach questions like this, and where do you think biology is going to land on the whole for some of these other indications?
That's an excellent question, and a question that it is a little bit hard to generalize, but I will try to discuss it a little bit. In a way, the trial that we started in when I was in France, and actually we published that a few months ago, is one of the examples. So Crigler-Najjar syndrome is actually a deficiency of a protein that is not secreted. And we know that heterozygotes are actually healthy. So we know that 50% is enough. But we also know that in a way with AAV, at let's say the doses that we intended to use, 50% transduction is very hard to achieve. And so we did a lot of experiments in case you have an animal model, let's say, you can do the real life experiments where you try to see if you can achieve correction of your disease phenotypes or biomarkers, and what you need to get to get that correction. Now, playing by simple numbers, so 50% or 30%, and using the genetics is not always exactly right, just because sometimes or oftentimes in gene therapy, you tend to have one cell that expresses more than 100%, right, of a wild type, because we use fairly refined and strong promoters to drive expression. And so what we learned in Crigler-Najjar is actually that we think that the level of expression that we achieved in liver are probably around 5 to 10%, so pretty low. But then the assumption is that probably these hepatocytes, they express more than a wild-type cell. And the clinical experience, as well as preclinical, showed that we could achieve in the case of Crigler-Najjar, is the clearance of bilirubin, unconjugated bilirubin from the circulation. So we saw that both preclinically and clinically. Again, here is a very good animal models, very good biomarker, so everybody's happy here. And that's not always the case, and then it can be more complicated. It's hard to generalize because in some cases you really need to hit probably most of the cells to achieve an outcome, because even having a cell that is not corrected may cause an issue.
Right. Yeah, it reminds me of, because I trained as a developmental biologist once upon a day, and it's that difference between sort of cell autonomously required in a tissue, where maybe 5%, as long as it's in every cell, is good enough, versus required more bulk action, if you will. So, if 5% of cells are 100% corrected, then that's good enough. It's a very different sort of challenge between those two for AAV.
I would say probably one recommendation to make it practical, I like practical things, is to try to develop some kind of a model that there is a disease model where you can have an idea of the ballpark or where you need to get, and try to mimic in a way or understand your efficiency of transduction and transgene expression in a model that is relevant, may not be the disease model, but is relevant to humans, like a large animal model.
Right.
And so I think that's very, very important. And it's something that becomes very important, for example, or particularly more important for tissues that are harder to transduce efficiently. I'm thinking about the brain, right? So you want to really optimize your delivery and your overall vector and everything that you do around it to really achieve the desired coverage, the desired transduction of your target tissue. I'm being a little general here, but I think it's very, very important to make sure that you have some data in a model that represents well the complexities of the human model, and that's not the mouse for sure.
Right. Right. Well, and I think, it's funny, sort of as you were speaking, I almost felt like it took us back up to the top of our discussion together because, you identified like having devices, for instance, that might help you to improve the exposure in the brain, for instance, having other technologies sort of wrapped into the sort of core of a capsid plus a payload, and how this, at the end of the day, all of this that we're doing is teamwork, because none of us is going to be an expert in devices and the animal models and the disease, perhaps, et cetera. And so I'm curious, maybe to return us back to the beginning. When you're sitting on a team who's struggling through one of these issues, how do you help that team get clear on a framework for figuring out how to take that next step? Like, what have you seen help teams sort of, I don't know, unhook and figure out how to even take that next step forward when they're faced with a lot of these questions and unknown unknowns as well?
That's really important. I would say the easy answer to your question is, and what I always do actually, is I help people go find the right competencies they need to get what they need to do. For example, I think if you're trying to develop a gene therapy and you're trying to bring it all the way to the clinic, then you need to have, for sure, very strong help. I'm going to call it help, right? Or competencies in the whole part that is related to the product. So not only the process development, so the manufacturing, but also the analytical. Because then you get to know what are you're actually putting into patients, which is crucially important. And then of course, depending on what you're trying to do, I think if you're facing some complex disease or CNS disease, you definitely need a neurosurgeon. So the key is you need to assemble teams with the right competencies, and there's those they don't sit in one person, 100%. And I've seen that in my previous job as Spark, where we truly did a lot of work over the years to have the right people around the table to develop the therapies and the right advisors outside to make sure that we covered all the complexities of what we're trying to do. So probably to summarize my answer, when I find myself talking to people, I try to peel off all the layers of complexity and try to tell them, okay, you can get this from here and the other from there, but it's hardly ever seen, let's say, one group that has it all, right? Because there's a lot of variables and a lot of things you have to take into account.
I guess one last question for you, and it's my favorite question. If you could wave a magic wand and fix one thing, actually, you're not limited to one, but if you can fix a thing that would help cell and gene therapy development, what would it be?
I love this question. I could probably name a few technical tricks that I would love to see solved. But actually, I think that the field is gone a long way and will get there. So I think things like cost of goods and manufacturing or immunogenicity, those things will come. And we're really truly getting there. What I would love to see is that the field and the regulators reach the right framework to help develop gene therapies for all diseases. In a way, we've seen in recent years that there's a shift of the industry a little bit away from rare, ultra rare, and so from diseases that don't really have a commercial value. And I think that's obviously sad, and they leave us with an important problem to solve, and then needs to be a combination, of course, of some technical advancements, but also some shift in the way regulators see the development of cell and gene therapies for those diseases. Otherwise, they will be always too expensive, too difficult, and maybe sometimes impossible to develop therapies for these rare and ultra-rare diseases. So I wish if I could just use my, if I was a magician and solve that, I would do it right away.
Right. Right. No, I like that a lot. And I think you point out something, incredibly important, right? Which is that the challenges of developing, forget even rare, ultra rare therapies are so difficult. And we are, as much innovation, I think, as we're pouring into this field technologically, we're going to need to pour into every part of what we're doing, be it regulatory, be it, the payer side of things. We're just going to have to be as innovative as we can in order to see the benefit for as many patients as possible.
Yes, and I think that there is a rationale, because we see and we've seen that gene therapy can actually make a big difference, can provide treatments that in some cases are curative for these diseases. So we cannot quite disregard that.
Right. Yeah, it's, first of all, I agree with you. I think, we as a field need to continue to sort of aim for that sort of almost curative or basically curative sort of targets for these indications. And the hope has got to be that we can find some innovative strategies to find paths for approval. If that's what we're delivering, hopefully we can find the regulatory flexibility to help that be successful.
Right, exactly.
Great. Well, Federico Mingozzi, thank you so much for spending time with us. Really appreciate it, and really appreciate everything you do. And I'll continue to hand out your 2020 molecular therapy article and drive up your score on that.
Thank you, Emily. This has been great. It's been a pleasure to have this conversation with you today.